So thanks everyone for coming to the talk. Let's welcome President James Liao for an opening remark.
Ladies and gentlemen, good afternoon. It's our great pleasure to welcome everyone to this Nobel lectures. And this series of lectures is organized by Academia Sinica along with the International Peace Foundation and a few universities in Taiwan. And we already have quite a few lectures. And today will mark our additional one with the laureate from Norway, Dr. Moser, who won the 2014 Nobel Prize in Physiology or Medicine. And Dr. Moser discovered the grid cell together with the plate cells, which was discovered a little bit earlier, form the basis, neurological basis for how we construct our cognitive map in our brain. And that discovery revolutionized our brain science as well as how we know about our brain functions. I want to mention one particular thing is that Dr. Moser was initially trained as a psychologist, and then he moved to experimental neuroscience. And with deep understanding, careful experimentation, he discovered this great phenomenon. So I'm sure you enjoyed his insight today, and hopefully through this interaction, we'll learn more about how great science is conducted. Thank you very much.
So thank you everyone to coming to Dr. Moser's talk, and I especially thank Peace Foundation for the generous support and the support from Academia Sinica. My name is Ching. I work for the IBMS and the Neuroscience Program of Academia Sinica. And I think that President Liao gave a very good initial introduction, so it can save us some time. I want to briefly talk about his Dr. Moser's background. He had his undergrad training in psychology and mathematics in University of Oslo. And then he went on to have his postdoc training, the PhD training with Per Anderson in the University of Oslo, a postdoc with Richard Morris of University of Edinburgh. And then before he started his lab in this new university, the Norwegian University of Technology at that time, he had a short stop at UCL in John O'Keefe's lab, who initially identified for the first time the plate cells from rodent brains. So as we know, he has serious discovery of not only grid cell as a quote-unquote GPS system in the brain, he also found many other different cell types. Hopefully he will mention some of them in his talk. And I also want to mention that he has a remarkable impact on many different fields. It's not only deep, but also wide. In terms of neurodynamics, the British discovery of plate cells from John O'Keefe and the idea of cognitive map in the hippocampus is a conceptual advance, whereas the Dr. Moser's contribution really bring the mechanistic perspective into this entire business of the explanation of what traditional psychology try to explain. In terms of memory, this foundation of a coordinate system become a basis where the experience of space and time can unfold. In terms of the navigation robots, for example, we all know robots need to be efficiently map the space, localization and mapping at the same time. And Dr. Moser's finding is an important biological implementation of that, which provide a lot of inspiration in robotics. And believe it or not, AI system built from the Google DeepMind also found plate cell-like correlate in their AI system when their agent need to navigate. So it has a lot of inspiration, provide a foundation for inspiration for today, what people talk about as a world model, that hippocampal grid cell system might be part of the biological correspondence. So there's one last thing I want to mention about Dr. Moser. When I talk to some very senior scientists in the US, they sometimes say Nobel laureates, some of them, impact or the citation would drop after they receive the prize. But what I found from Dr. Moser is exactly the opposite. He is very focused, and he continued to contribute to some of the even more important findings after he received Nobel Prize. So I think he will be a very good role models for many of us as what a scientist should be like. So let's welcome for his talk and enjoy his talk. Thank you.
For these general words, thank you also for the introduction to President Liao. So I appreciate the invitation, the opportunity to present some of the work we are doing at the lab in Norway. Some of you may have heard some of the work from Edvard Moser, who spoke at the university, I think, a month or two ago. And but I hope that there's still something novel to tell. And the focus of my talk will be on our sense of space, mostly. That means, how do we know where we are, and how do we get from one location to another? And if I have time, I will also touch on our experience of time. It's another fundamental cognitive function that we have. But I want to step back, since this is not only a neuroscience audience, but also a general audience, and ask some of the questions that have been around since the earliest days of philosophy. That applies to Greek philosophy, but it could also be a philosophy in the East or other places where people have asked about the origin of our minds, the origin of our thinking, our thoughts, our feelings. Where does this come from, and how does it work? And this is the origin of philosophy, which then has arisen many places in the world, but turned into a more systematic inquiry with the birth of psychology, which emerged only in the late 1800s, at the turn of the century to the 1900s, when independently, or also to some extent dependently, investigators like Wundt, Helmholtz, Freud, James asked questions about our mind, and what can we learn about our mind, what methods can we use to learn about the mind, and where quantification was introduced as one way to do it, for example, in Helmholtz studies of memory. But the underlying factor here is, of course, the brain. It's the brain that makes us have thoughts, have feelings, have plans, have ideas. And the brain obviously has not really been accessible due to the lack of methods for doing it. And this is this is a discipline that grew in the 1900s, but really took off only at the end of that century, and now in the 2000s, where new methods have made it possible to investigate the brain and actually relate it to these functions that philosophers the questions that philosophers asked thousands of years ago. So, but still, nonetheless, and throughout my lifetime, when I started out as a psychologist, there has always been this perceived gap between mind on one side and brain on the other. So, how can we bridge? How can we create a science that actually links what we perceive in psychology with what physically goes on in the brain? I'll start this video, an illustration of what the brain looks like, right? So it's in the human brain, almost 100 billion neurons, each with connections to about 10,000 other cells. So here you see typical cells, what they look like, and they have these connections which called synapses, where electrical activity in one cell leads to the transmission of chemicals called neurotransmitters to the other side, which then causes a new electrical signal. So this electrical chemical communication is the brain. That's the language of the brain. That's how it works. But it is distributed so over all these 10 to the 11th of neurons or 10 to the 15th of connections. So how can we find the information in the brain when it's so small, so distributed? It's really a huge task. But there has been progress, and in some areas it's easier than others. And one of those areas, when it comes to cognitive functions at least, is our sense, quotation marks, of space and time. So these are brain functions that are very fundamental to our existence, and so fundamental that it has been proposed by philosophers like Immanuel Kant that they actually are part of the brain. You cannot imagine, for example, what space is like, with what the world is like without space or without time, because we perceive the world through our perception of space is really formed by our brain. And what is outside there, not perceived by our brains, that's actually very hard to imagine. We can quantify it in physics, but still our daily perception of space and time, that's very much shaped by the constraints of the brain. But that also is a good thing, because it allows us to study those constraints and maybe learn something fundamental about how brains work. So then, focusing on space and to a lesser extent time today, I would start by claiming that these are so fundamental that they must have developed very, very early in evolution. So because without those, there's hardly any chance you can survive, really. So if you didn't have the sense of space or time, you would certainly not find food. You wouldn't be able to escape from predators, or you wouldn't find a mate. So nothing of all these things that are needed for survival would really be there. So obviously, this must have been developed very early. This shows a phylogenetic tree for mammals, and you see humans branching up at the top right there, and then you have rodents to the left, and you have monkeys, and you have bats down there, and so on, and you have the common ancestor to the at the bottom left. So this is an illustration from Liao Krubitzer. The idea here is that since our sense of space and time evolved so early, it's probably very similar across species with certain adaptations, but nonetheless the general topics have been preserved, and everything points in that direction. In fact, many of the mechanisms probably even evolved much earlier, so that a sense of direction can even be seen in flies. In simple fruit flies already have some sort of compass-like system. So that just shows how early this probably evolved, because it is so fundamental. But my focus today will be on the mammalian brain, and I will begin and spend almost all of the time on space. And then ask, this is a human brain, then which brain areas are relevant here? And of course, in some sense, the entire brain is involved, but there are some areas still that are more critical than others, and I show two of the structures here. So one is called the hippocampus, which is shown in red, and then another one is called the entorhinal cortex, which is shown in blue. And those are located in the medial temporal lobe, that's behind your ears, kind of, on the still cortex, the sort of intellectual part of the brain, but it's more on the bottom and medial or middle side of the brain. So it's not on the surface like it looks like here, but since I don't have a 3D image here, then you imagine this is a little bit into the picture. All right, but studying human brains is not an easy thing to do. there are studies of human brains, even of single cell studies, but usually in connection with work on patients who have, for example, serious degrees of epilepsy, and then they have electrodes in the brain where you can pick up the electrical activity and study the signals there. But still, if you want to have single cell resolution in the work, then it's easier to go to animals, which anyway have much of the same circuitry. So this shows the two key brain structures of my talk, namely the entorhinal cortex and the hippocampus in a human brain at the top, and at the bottom you have a rodent brain. It could be a rat brain, but the mouse brain also looks not too different. You can see that those two structures are identifiable in both species, and if you look at the internal organization, it's also very similar. The same subregions called CA1, 2, 3, and then the gyrus, same here as well, and then those connect to the entorhinal cortex. In many ways, it's a kind of information circuit where information goes from entorhinal cortex, goes through a circuit in the hippocampus and gets back again. So I won't focus much on that, but let's just keep in mind then that the circuit is quite similar, which allows us to infer very much from the rat brain to the human brain. So going back in history, then one of the first things to notice, one of the real milestones was the suggestion that there are maps in the brain, that our navigation actually is based on maps. This is the work of Edward Tolman, who worked at the University of California at Berkeley in the 1920s, 30s, 40s, and 50s. And he proposed, based on studies in rats, which walked in mazes, for example, that the brain, both of humans and of rats, have internal maps. He suggested that based on experiments where he noticed that the rats are able to make shortcuts or detours. That means that if they're trained to go from A to B to C to D, then they can actually make a shortcut from, let's say, from A to C, if that opportunity comes, instead of just walking the path that they have been trained all the time. So we make shortcuts like that all the time, and it's the most obvious thing we can think of. But at that time, it was quite controversial. So many psychologists, experimental psychologists, didn't really accept this new conceptualization that we actually have maps of our surroundings. And not only did Tolman suggest we have maps, but he even suggested that maps are part of knowledge structures in our brain, where we connect the physical environment with information, knowledge we have about what's going on in the environment. And that knowledge structure he called cognitive maps. So this remained controversial, but then it did help then when 30 years later, John O'Keefe started to record from the hippocampus of rats, which were doing various tasks like walking in mazes or walking in open environments. And what he found was the following, which I will show on the next slide. So what he did then, first, before we go to the next slide, he implanted very thin electrodes, sensors, into the brains of the rats, and then for the occasion in the experiment, connected those sensors through a cable to a computer, and could then record the activity and the signals here, or electrical signals that we call action potentials. And they could do this while the animals were navigating around in the environment. So I'll show such a task here. So what you now will see is a video that I will start very soon. But you will see a rat walking around in a box that has a size of 1 by 1 meter. And it's picking up small pieces of chocolate. So this is a trick we use just to have the rat walk around and visit every possible place. And you will be listening to the activity of one single cell in the hippocampus. Each time the cell is active, you'll hear a pop, pop, pop sound which is when the cell fires so-called action potentials. So let's just do that now then. So this is the activity of the cell, right? So this is the action potentials of the cells. And each time the cell is active, you also see a red dot appearing on the screen. And what you now can see is that this cell is active only when the rat is up in this top left corner of the environment. Otherwise, the cell is electrically silent. So you notice here, and O'Keefe noticed that the cell is active in one place, and for that reason he called this a place cell. So other cells were active in other places, so showed here in a color-coded version. So red color means high activity and blue means low activity. And you can see the different cells are active in different places. So this fact that different cells are active in different places, that led John O'Keefe and Lynn Edel. I understand Lynn Edel has been here and talked, so that's also interesting. This picture was from 1975, so it's quite a while ago. But both John and Lynn are still very active in the field. And what they suggested, based on these observations, was that the hippocampus is actually the place cells are the cognitive map that Tolman proposed many decades before. That means that it serves as an internal map, and depending on which cells are active, then you can read out from the brain where the animal thinks that it is. So this was in the 1970s, and then in the 80s, 90s, another type of cell was discovered. So these are the so-called head direction cells that were discovered by Jim Rank to the right here, and with Jeff Taube, his student at that time. And what they found was cells that are active in a certain direction, not in a place, but when animals face their head towards a certain direction. So these cells were first discovered in this yellow area here called the presubiculum. And it's sort of squeezed in between the hippocampus and the entorhinal cortex. Recordings in that area showed what you see at the middle here. This is from the original paper in 1990, that this particular cell is active only when the rat has his head turning in a certain direction, which in this case is relative to the room, 270 degrees approximately. Otherwise, the cell is silent. The other cells are active in different directions, and together you have just like place cells form a place cell map, these cells form a direction map. And if you read out which cells, directional cells, are active at any given time, you can tell which direction the head is, the rat is facing. But then direction and place should work together if they are part of the navigation system. And that's where leads us to our work. We're in our lab, where the work that I have done together with Edvard Moser, we started to record in the early 2000s from the third area here, namely the entorhinal cortex. So this is the blue area shown here in the rat brain. And to the left, you see a rat walking around in a box, just like in the place cell video that I showed a few minutes ago. White is when the cell is active, and you can see that this particular cell here from the entorhinal cortex is active in certain places, just like the place cell in the hippocampus, but it's active in many places, clusters of places, and the cluster of places, they form a certain pattern, and you can see the pattern to the right. So to the right, you have the trace in gray, you have the trace of the rat where it had been walking over a period of 30 minutes. And each black dot shows a place where the cell has been active. And what you can see is that those activity places form a regular pattern that repeats over and over and over again in the entire space. Kind of formed like equilateral triangles, or if you want a hexagon, or if you want a rhombus. So, but anyway, it's the same pattern repeats all over again. So because this is like a grid that covers the entire environment, we call those cells grid cells. And it's also important to say that grid cells come in different varieties. So different grid cells may have a different phase, first of all. So here are two grid cells, one in blue and one in green, and you can see that they are shifted in the XY space. Or they may have a different scale, so you have different blue and green. The blue one here has a bigger scale, so it's wider fields and longer distance between the fields, or the orientation might be different, means that they are tilted differently. So all of these cells exist in the system and together form a map that exists at different spatial scales and also covering the entire space by being distributed across the full XY space of the environment. In many ways, we have never really said that this is the brain's GPS, but that's what people generally said. They even said it when they read up the popular version for the Nobel Prize, that it said that this is the brain's internal GPS. And the reason is, of course, that this can serve as a navigation system. So it's still quite different from satellite-based navigation system, but it is similar in the sense that these cells track our movement in space, and depending on where you move, in which direction and how fast, you have different cells that are active, different grid cells activated depending on your movement. So in that way, you can infer from your movement where you are in space. You don't really need to know anything about the landmarks. And that's just what is similar to the GPS, that you don't need really to plot in houses and trees and rivers or anything. You just need the XY coordinates and that's it. And in that sense, you can perceive this also as a kind of coordinate system that the brain uses. So this is where we were until some years ago, but now things have changed very much. And over the last 10 years, one of the big changes is the introduction of new technologies for recording not one cell at a time any longer, but rather from thousands of cells at the same time, and then asking how do they work together. So the transition has been from single cells in a way to populations. And what you see here is, first of all, to the left, you see a new type of probe, which is really just a microchip. It's a silicon probe that where what goes into the brain are these 4 long shanks here, or which actually consists of it's like a chip where you have thousands of recording sites on a strip, and then you have 4 of them, and that gives you 5,000 recording sites instead of just 1 per electrode as you had before in the old days. That makes it possible then to record activity from thousands of cells at the same time. So this shows just 149 of them. They are all grid cells belonging to one particular module or size of grid cells. And then you have time on the x-axis, and then black means active, and white means they are not active. This then, the fact that we can record the activity of so many cells, each row here is 1 cell, that means that you can study the combination of activity. And the combination of activity is really what gives the unique information that happens at any given time. But then, imagine how many combinations there might be. So if you now have just 149 cells, and they're all independent, then you would have to plot them in a 149-dimensional coordinate system where activity for each cell would vary along the axis that you have in this coordinate system. Imagine then that there are 10 levels of activity on each axis. That means that you can, in principle, get 10 to the 149 combinations, which is more than the number of atoms that you have in the known universe. So it is so many possible combinations that it's actually not only hard to imagine how it could work, but also hard to work with as a scientist. But luckily, the activity of these different cells is actually very highly correlated. So you don't fill up all possible spaces in this in this space at all, not really at all. You can actually break it down by so-called dimensionality reduction techniques to explain most of the variance in just a few dimensions. For example, plotted in a three-dimensional coordinate system, which captures most of the activity because so many of the cells are correlated with each other. And that introduces us to a new world. So again, then here you have the original raster plot. So in this case, 149 cells. And you break it down to the key components. So for example, 3 that you plot, now the evolving activity over, let's say, a period of 5 seconds, then moves in this three-dimensional space here. As for example, if a rat moves around in a box, in an environment, you can see activity in the map, whatever it looks like, evolving around because the combination of active cells also changes. And when we do that, then the following happens. This is what we get. So what you have here is one dot in this point cloud, one dot per time frame. So remember the columns here. So each column is the combined activity, but now broken down and shown in a three-dimensional embedding instead. And then each time point is one dot, and all the time points you recorded over, let's say, half an hour are shown in this cloud. And as the animal walks around in the environment, you also have the activity moving around in this cloud of possible spaces where the activity could be. And as you can notice here, this point cloud, this space has the shape of a torus or a donut. This is in many ways, this is the total activity you get if you record over a time when a rat is walking around in an environment. So this, in many ways, is the internal map of space. All the possible places where an animal could be is shown here. It has the shape of a torus, and this is in some sense is Tolman's internal map, because as we move around in the physical space, is always matched by some position in this toroidal map in the brain. And what's quite striking is that this map even exists when we sleep, when we do nothing, when we rest, REM sleep, slow wave sleep, any kind of state, the activity still moves around on this map, even if you don't move around in physical space. So this suggests then that this map, the spatial map that we have in our brain, which is the basis for the grid cells, is actually quite independent of behavior, and that activity moves around on the map even when we are just doing nothing. So this is consistent with theoretical models of how grid cells could operate, where the idea has been suggested already in the 1990s that you have different cells, so each cell is a dot here, and then the color indicates the activity of the cell, and this is each diagram here shows the total activity in the brain network. And then you can see that the activity moves around in the network in correspondence with where the animal is in its environment. But if you have a flat map like this, you get one problem. What happens when you get to the end of the world? So when you get to the end of the map, do you fall outside the map? No, you don't. And the solution is the same as occurred to scientists some centuries ago, that the world is not flat. It's actually is periodic. So if you have an internal map that is, for example, a torus, you will never ever get to the end. It will just go around and round and round. And you just need to link it to the external environment, but the map has no end. So that's an obvious advantage. And it also then relates the question which I started out with, namely, is this an internal map that actually is from the beginning? Is it a priori, as Kant suggested? And the question then is, were we born? Are we born with internal maps of space? Are we born with these skills, or do we learn them by experience? And this relates to discussions that have been around in philosophy for centuries, whether all our cognitive functions are actually learned, or whether, in other words, whether we start from a blank spot where you have nothing and then everything we know is learned, or if you actually are born with ideas or mental functions or models, what do you call them today, that we come to the world with prior to any experience. So Matteo Guardamagna in our lab has tested this. It is now possible to record from young brains with these neuropixel probes. So here you have the timeline of rat life, starting from postnatal day 0, when they are born, to shown up to 30 days here. And most of the work that we had done previously is done in this yellow time window here, from the day postnatal day 15, which is the day when rats start walking and leaving the nest where they are born and start walking out exploring the world. And then in that early work we found that there are cells that look like grid cells. They're very noisy though, but they're already there. And that led us to ask then, how early can you actually have these grid cells around? But what was possible now with the neuropixel probes and with the technical skills of Matteo was to start recording from rats that are as young as 8 days old. When at that age, they haven't opened their eyes, they have not opened their ears, and they don't walk. So it's a very, very immature state for a rat to be in. So they're blind, they're deaf, and they can just slide a few centimeters inside the nest just to feed, and that's all. Most of the time they're sleeping, 80 to 90% of the time. And Matteo was able to record from brains when rats as young as 8 to 12 days were sleeping in a pot. And what he found is this. So you can certainly recognize the torus here. So in this experiment, it wasn't possible to pick out grid cells because we can't record grid cells at that age because the animals don't move in the environment. But what he did was that he took all the cells in the recording, 600 plus something, and recorded, correlated all of them against each other. And then if you do that, you find that some cells are highly correlated with each other, and they form blocks or squares in this cross-correlation map. So if you take those cells that are highly correlated with each other, like the yellow one here, or the one in the middle here, or the one here, and then take each of them and then plot the space over which all of the activity is distributed. So this is the so-called UMAP space. Then you see that it has this characteristic torus-shaped nature. And you can even then find that every cell has its own distinct location of activity on this torus. So you just take the torus here and unfold it to a rhombus, and then you find that different cells are active in different places. So this means that this internal torus, the map is actually already present before any experience with the world. The structure is already set before experience. Then it turns out if you now do the same for different days, day 8, 9, 10, 11, and 12, and so on, you see that this transition actually happens on day 10. This is the 10th day of rat life. Then suddenly, if you measure the degree of torusness, then you can develop a score, a toroidal score, and you'll find that when each dot here is one experiment, or one module of grids, or module of cells, one cluster here, you'll find that those with a high torus nature, they appear only on day 10 and then onwards. They are not present before. So something really magic happens on this day 8, and then you may wonder, what is it? If you then examine the network activity of the cells on day 8 and 9 versus day 10 and 11, you can see it's very, very different. On days 8 and 9, then you can see that the activity is very burst organized. So that means that as time passes, so in this case it's 5 seconds, you can see that all the cells are active at the same time, and they're probably triggered by influences from the outside, like muscle twitches, or it could be some other sensory input. But this differs very much from what you get from day 10 onwards, when it gets very noisy and decorrelated and the cells are active all the time. So it's a switch from a very externally driven activity before day 10, and after day 10, it's much more, the cortex is much more self-going, and it's active all the time, probably reflecting the fact that you get connections between the cells, which is a condition for actually creating such internal maps. Okay, I skipped that one, but let me now just spend the last part of the talk on the dynamics of grid cells, because what I've told so far is about the static map-like nature of our internal maps. So what you see if you average over time. But actually, if you are to use the maps in the brain for something, you need to update them second by second, or even millisecond by millisecond. So there's a fast dynamics here that we haven't been able to study. And this is important because if you use these maps to find your way, you actually need to relate your current position, where you're just now, to any future position where you want to go. You have to know how to get from A to B. It's not sufficient to know where you are. You need to know how you can get to other places as well. So how is that done? And this is the work of Abraham Wollan in our lab, which it's still based on these neuropixels probes, where you can record thousands of cells at the same time, but now at a time resolution of 10 milliseconds. So for every block of 10 milliseconds, what he did was that he decoded from the combined activity where it's most likely that the rat is at any given time. So he first created maps of found that where every cell is active, so that he could then infer from the combined activity what is the most likely position that the rat is in at any given time. Then he does that because he now has so many cells that he can ask for every 10 milliseconds block, where is it most likely that the rat is? And then you can see that the grid cell map is not static, it's actually changing over time. And I will now play a video that illustrates that. So this is slowed down very much, but you can see that the activity, which is color-coded here, goes to the left, to the right, to the left, and to the right, and to the left, and to the right. And these left-right sweeps, they happen every 100 milliseconds. So each sweep is left-right every 100 milliseconds, and the color just illustrates time within this sweep. So as I said, at steps of 10 milliseconds. So that means that the cells, the grid cells, and also the same for the place cells, they don't really encode they don't only encode where the animal is at any given time, as people thought before. But if you break it down into very high resolution, it actually encodes future places, which then are systematically mapped from the left to the right, to the left to the right of the animal as it's walking forward. And you can see that in the summed up in the diagram here, where every second is left, and every second sweep is left, and every second sweep is right. Okay. So where do those sweeps go? It turns out that those sweeps don't only go to places where the animal actually ever will go. So this shows when the animal walks on a maze. It's a circular maze, and you can see the sweeps go left and right and left and right, but into places where it's not physically possible for the animal to walk. The same happens when the rat walks in a box. The sweeps may even go outside the box. So, and even when they are sleeping in REM sleep, you still can reconstruct such sweeps that go left, right, left, right. So that means this is very much hardwired. It's really an intrinsic part of the brain. So what is intrinsic is not only the torus-shaped nature of the map, but it's even this exploratory left-right pattern that happens every theta cycle or every 100 milliseconds. And moreover, it's not only the grid cells and places that sweep to the left or right, but it actually turns out there's a separate population of directional cells one step upstream in an area called the parasorbiculum. And those are head direction-like cells that also go in the left-right pattern. You see it with the green arrow here. So they are totally synchronized with the sweeps in the grid cells here. And the sweeps are the color-coded ones, and you can see the directional signals just go left, right in the same pattern. So what could such a pattern be good for? So I'll continue about 5 minutes and I'm done. But what is such a pattern good for? So this might be an efficient way to cover space in the environment. So it's actually, if you do this left-right alternation and the rat moves forward, by alternating this way, it's at a certain angle, which has an angle that's usually 30-40 degrees, then it's actually a very nice way to cover the upcoming space. But sometimes not all spaces need to be covered. Sometimes there are some places that are more important than others, like, for example, when the rat moves to a certain goal, or when it wants to get around an obstacle, like a wall, or when it, for example, wants to chase some bait. So I'll show now an experiment where a rat is actually chasing or running after a piece of food, which is on a fishing rod. And you can see the fishing rod or the bait is in green, and the rat is running behind to try to catch it because it likes it so much. And this is a real recording, so you can see the bait and you see the rat coming, trying to chase it to get the food. So we use that task as a way to ask, do these sweeps actually adapt to the specific environment? And it actually does. So now you can see the sweeps actually are much narrower, they're much more focused on the bait. And so when it's something important that happens in the environment, then this sweep system actually doesn't cover the entire environment, but are much more narrowly focused on the goal. So the goal still go left, right, but the angle is sharper and is much more focused. And this shows an example when the bait suddenly goes behind the animal. Now you can see that before the animal turns, the sweeps actually go back and follow the bait. So look again, once again, now the bait goes back, and then the sweeps go back, and only now does the rat turn. So that is actually ahead of the behavior. So there's a kind of mechanism. These sweeps are actually prior to behavior. So it could serve as a way for the system to monitor the environment and the relevant parts of the environment, which then may give rise to the subsequent behavior. And with that, I think I could have said much more about this. This is what we're really working on. But to sum up about the sweeps, then I will just emphasize sweeps as a pre-behavior look-around mechanism that where place cells and grid cells sample the environment beyond where the animal actually is at the current time, and that it serves as a spatial focusing mechanism to sample parts of the environment that are particularly relevant. So with that, I will, before now, I had planned to say a few words about time, but I will skip that in the interest of having time for questions. But I want to have 2 slides about the links to disease and neurodegenerative diseases, and particularly Alzheimer's disease. So it is, of course, noticeable that Alzheimer's disease begins with degeneration in the very, very same areas of of the brain, which is entorhinal cortex. So among the very first places that cell death starts in the Alzheimer brain is in entorhinal cortex. There are a few others that are also very early, but this is certainly among the earliest. And This shows here a normal brain here, you have the cells here, you have a mild cognitive impairment, which is called, of course, kind of pre-Alzheimer, and then you have Alzheimer here, and you can see the thinning of cells here, and there's also a lot of pathological markers that go along with it. But an interesting observation is that although there is no cure for Alzheimer, then this degeneration in the entorhinal cortex, shown here by color code, especially in the lateral part of entorhinal cortex, begins many, many years, decades perhaps, before the full symptoms of Alzheimer's disease break out. So that gives us at least some hope that when a drug or some treatment is developed, then it will probably gives us some time to interfere. And of course, this goes along with spatial disorientation and loss of memory, because these brain areas are so important for that function. And I don't have to tell, because everyone knows that Alzheimer really increases with age, and with the world getting older, then life expectancy is increasing. Just this statistic over 15 years, it's 4 or 5 years, in most of the world, all over the world actually. So that just shows that this is becoming a really big problem all over the world. But Alzheimer's is not really the primary target of the work that we are doing in our lab, but it's a certain implication. So hopefully this concept of the focus of primary research and basic research and clinical research will one day solve this puzzle. So with that, I just want to show the picture of the people involved in this. This is the research group. I mentioned many of them before, but I mentioned Matteo Guadamagna, who did the development work. Abraham Vollan was not here. Edvard Moser is there, and the others have also participated in many. It's a team of people from all over the world, and I think with that, then I didn't get time to say much about time, but we have also worked on that, and that is time is much less understood than space. But the one-line summary of my time section is that in lateral entorhinal cortex, but actually in the entire hippocampal system, there is a constant drift of activity that correlates with the passage of time, and especially when important things happen, so that this may be a basis that the brain uses for inferring how much time has passed. So with that, then, thank you for your attention, and I would be ready for any kinds of questions that come up. Thank you very much.